Question 10. Okay, question 10. Last one that we're going to look at, because I'm not looking at question 11, or indeed, was it 7 or 8, whichever one I didn't do as well, because that's the extended answer, and I don't think that I am adding any explanation to what's in the mark scheme. Right, question 10. The figure shows an Ebola virus, cause of the Ebola virus disease, EVD, prevalent in a number of West and Central African countries. The virus is transmitted to people from wild animals, and outbreaks may then occur through human-to-human transmission. Ebola viruses have a diameter of 8 times 10 to the minus 2 micrometers. Give this measurement in nanometers. 1 nanometer is 10 to the minus 3 micrometers. So you have, be careful which way you're doing this round. So the nanometers are smaller than the micrometers. You have 1,000 nanometers in 1 micrometer. Okay, you're told that it's 0.08 micrometers. So I just need to times that by 1,000. So I'm just going to jump my 1, 2, and then a third for there, and I get 80 nanometers. That's it. Okay, B, I'm up on the right here. The flow diagram shows some stages in the replication of this virus. The virus attaches to host cell receptors, it enters the host cell. Viral RNA is used as a template to synthesize complementary mRNA. mRNA is translated into viral proteins, viral RNA is replicated, new virus particles assembled, virus is released and cells destroyed. Explain why the Ebola virus cannot be regarded as a retrovirus. Okay, retroviruses go in your various stages of, okay, here's your cell, here's your virus attaches. The virus has to put in RNA, which is then reverse transcribed with reverse transcriptase to make DNA, and then that DNA is then inserted into your host chromosome set, host genome, we'll say. Okay, and then at that point the DNA is then transcribed to mRNA and then goes through the whole cycle, right? This just isn't doing that. So we have the viral RNA is used as a template to make RNA. So we have not got reverse transcriptase, we have not got RNA converted to DNA, and we've not got it being inserted in the host chromosome. So you have all of those are reasons to say it's not a retrovirus. Okay, right, sorry, I spin it down a bit for C and down to F. Right, EVD symptoms result from viral disruption of immune cell functions such as the loss of lymphocytes by apoptosis. State the cause of cell death during apoptosis. So this is caspase action. And that's basically what we're saying. So caspases destroy their proteases and they digest proteins. The genes coding for some Ebola virus proteins have a high rate of mutation. Why would this make development of a vaccine more difficult? Again, we're all up on this because of COVIDness. So basically, high rate of mutation means that the vaccines might not remain efficient or effective at picking up the differences that you now have. So for every new mutation that you get, you either have to redesign an entirely new vaccine, or you have to hope that it's not actually the bit that the vaccine was actually attaching to that has been altered. Okay, EVD has a very high mortality rate. Some researchers have suggested that new treatments should not be assessed by clinical trials that use a negative control group. State whether you agree or disagree with the suggestion and justify your answer. Okay, so whether you believe in almost pure science or whether you don't want people to die. So you could go with the I disagree. I feel that we should always use control groups, because if you don't have control groups, no control means that your conclusions are not supported. Your conclusions are not as yes, true, valid, supported. Okay, that is, however, pretty nasty, because basically what you're saying is that with a very high mortality rate, you are saying my control group, a lot of them are going to die. So what I'm expecting is that you're more likely to say, no, I agree with that, because very high mortality is unethical to allow. Like, you wouldn't want to be saying, well, it's okay, we'll just have a group of people that die. But as far as the mark's concerned, you put whichever one you say and then you explain why you're happy with it. So, yeah, that's your 2. Apart from medical treatments, suggests one measure that could be effective in controlling or preventing outbreaks of EBD in the tropical regions of Africa. Right, if I go back up a wee bit and we have a look here, it tells you something about this which will automatically give you nice ones. Okay, the virus is transmitted to people from wild animals. So there you go, there's something you could say. So you try and prevent contact of people with wild animals, and that could either be by reducing contact, so either barricading effectively. Education would be useful to mean that people are not having contact with them, and that would help. Or you could say that you start treating animals, so preventing that being an issue for anyone who comes into contact with them. But it then says may occur through human to human transmission, so we then need to cut down human transmission. So best thing to do that is probably to reduce overcrowding. Okay, the less people you have together, the less chance you have of human to human transmission. And again, education of what that is. And again, we are so, or should be so, up on that from a COVID point of view, because everything we've talked about for the last 2 years has been about reducing transmission, and has become more and more clear as we've understood exactly how transmission occurs. And that's the question.